Well, good morning, everyone, and welcome to the Garvan Institute of Medical Research. It's an absolute thrill to have so many people here in person, and I believe we also have at least a couple of hundred people online. So welcome to our public seminar, Transforming Immunology, a cause that's very close to my heart, and I'm sure many of yours as well. It's my humble honour to acknowledge the traditional owners of the lands on which we meet, the Gadigal people of the Eora Nation, and pay my respects to elders past, present and future. We acknowledge their ongoing connection to land, waters and community, and recognise their more than 65,000 years on this beautiful country that we have the privilege of living on. In terms of some quick housekeeping, in the highly unlikely event of an emergency, please follow my instructions at all times. The exits are at the front of the auditorium, also at the rear of the auditorium, and if you need assistance, we have lots of friendly Garvan team members here to support your exit. Well, you're in for a real treat. We've got two incredible excuse me, asthma, topical. Okay, sorry, I've just got a little bit of asthma happening, which is actually, yes, very important. We'd love that to be cured, please. Elissa, thank you so much. I did just have my medicine before I got up here, but clearly I haven't had quite enough for the allergens that are in the air. So we have two incredible scientists and a scientist clinician joining us to talk about the immune system. And the immune system is really incredible. It can save us from infection. I might actually have to grab my Ventolin. Isn't that embarrassing? Maura, do you mind just passing my handbag? Sorry, everyone, I've managed to make this about me. How embarrassing and how quite silly. Excuse me, this is not part of the performance. My apologies. So, back to where we were. So, our first speaker today is Associate Professor Elissa Deenick. Elissa is one of our most senior female scientists here at the Garvan, heading up one of our core strategic programs, our Precision Immunology program. And Elissa is also chair of the Garvan faculty, which is a very important role here, mentoring the next generation of scientists. We will then be joined by the incredible Dr. Shruti Swamy, who is a clinical immunologist and a scientist working alongside Elissa, and she's going to share some of those real-life stories that impact so many of us. We will have a Q&A at the end, so if you think of any questions throughout the presentation, please do try to retain them to memory or jot them down, and we'll try to get to as many of those questions as we possibly can before we wrap up. I am just going to point out that I think that we might have some mums and dads in the room. So welcome to the Deenick family, and also welcome to the Swamy family who are joining us online. You must be incredibly proud of your absolute superstar daughters. So thank you for being here. So without further ado, Elissa, please come on up and tell us all about the immune system.
Thanks, Mara-Jean, and thank you all for coming along today, both in person and online. I'm really excited to talk to you today about how we're working to transform the way we treat immune diseases. We're going to go from rare diseases to common diseases. We're going to look inside immune cells a little bit and see how they function. And hopefully I can show you that there's a better way that we hope to find in the future of treating these diseases. But before we talk about immune diseases, let's talk a little bit about a healthy immune system and what it's meant to do. Our immune system is there to protect us from infection. Every day we're exposed to lots of things in the environment, viruses, bacteria, parasites, fungi, and the role of our immune system is to fight those off and make sure that they don't make us sick. And most of the time, our immune system does this so well we don't even notice. There are lots of things in the environment that would make us sick if we didn't have an immune system, but our immune system fights them off before we even notice they're there. And sometimes, of course, we get sick, but our immune system kicks in and we very quickly get better. So what is our immune system? Well, it's composed of many different cell types. Sometimes we collectively refer to these as white blood cells. They're the bit in our blood, if you separate it out, they kind of look whitish. But there's lots of different types of immune cells and they have different functions. And that's because the job that they have to protect us from all these different types of infections is complex. They need to be able to detect when these things come in, whether they come in through our skin or through what we eat, and they need to be able to fight them off appropriately, because the way that you fight off a virus is different to the way you need to fight off a bacteria. So you may have heard of some of these different immune cells, T cells or B cells, and we'll get to that a little bit later. So that's what a good functioning healthy immune system looks like, but sometimes it goes wrong. And one of the most obvious ways it goes wrong is when it can't fight off infection properly. And we call this immunodeficiency. And this can lead to severe or chronic infections. So for example, we might get a child who keeps having to be admitted to hospital with pneumonia and being treated by antibiotics. So this is, one way it can go wrong. It's kind of not good at fighting off infections, but there's other ways it can go wrong as well. So for example, your immune system can get confused, and rather than responding to and fighting off foreign things like bacteria or viruses, it can start attacking parts of our own body, and that leads to autoimmune diseases. So you will have heard of many of these: lupus, multiple sclerosis, Hashimoto's, scleroderma, and they're all attacking different parts. It's your immune system attacking different parts of your body. So think of any part of your body, there's probably an autoimmune disease associated with it. But there's another way, as Mara-Jean has demonstrated to us, that your immune system can go wrong, and that's allergy. So this is when our immune system isn't reacting to us or to viruses or something, but it's reacting to things in the environment that aren't really dangerous, like peanuts or pollen, but it overreacts to these, and this causes damage to our bodies as well, and the symptoms that you see of allergic disease, like sneezing, blocked nose, or anaphylaxis, which can be life-threatening. Now, to treat these diseases well, we need to understand not just what the symptoms are at the end, but what are the drivers, what's going wrong at the very beginning in our immune system to make it make these mistakes that end in disease. Now, to study that, for many years, I've been studying particularly immunodeficiency and people who are susceptible to infections. And there are many ways you can end up with immunodeficiency. There are drugs that do it. HIV can do it. But I've been studying patients who have genetic causes of immunodeficiency. So these people have a change in one part of their DNA, and as we know, the DNA is the instructions for how our cells function. So if they've got a change in the instructions, that can mean that the cell no longer functions properly, and they therefore can't fight off infections. So you may already have heard of these kind of genetic immunodeficiencies. Probably the most well-known person with this was this boy, David Vedder, who was also known as the boy in the bubble. So in the 70s, he was born and he had one of these genetic conditions, which meant pretty much that he had no immune cells at all, which meant that anything that he was exposed into the environment could have made him sick. So doctors, from the moment he was born, put him in a plastic bubble so that he wouldn't be exposed to those kind of things. So that's one form of these genetic immunodeficiencies, but there's actually a whole lot of them, and not all of them wipe out the immune cells entirely. Some of them just impact certain immune cells, and some of them don't make you susceptible to everything. They might just make you susceptible to viruses or bacteria. But it's really important if we want to help these people. These are rare diseases, but these are very impactful diseases. So for some of these people, if they're not treated, they'll die very early in life, or they may just have chronic infections throughout life. But if we want to help these people, we've really got to understand how these changes in the DNA are impacting their immune cells and how that then makes them susceptible to infection. And today I want to tell you a story of just one of these conditions that we've been studying, and it has a bit of an unwieldy name. It's called activated PI3 kinase delta syndrome. but we can call it APDS for short. So patients who have this condition get frequent bacterial infections, which can lead to pneumonia or frequent ear infections. They also get severe or persistent infections with some viruses, not all viruses, but some, including EBV, which you might have heard of because that's the virus that causes glandular fever. But these people get it and they can't control it, and it makes them very unwell. So quite a number of years ago, me and some of the other researchers here at Garvan were working with collaborators in the US, and they thought they'd found the change in the DNA that was responsible for this disease. And really then the question is, well, if we know that there's this change in the DNA, how does that impact the immune cells and why are these people getting sick? So we set out to really understand that. But it's actually a little bit more complex because it turns out there are two forms of APDS, APDS1 and APDS2. And they're caused by changes in two different bits of DNA, or what we call genes. So I'm going to have to, for those of you who don't really understand DNA and genes, I'll just give you a little explanation. I told you that DNA is the instruction manual that helps build our cells. And the way that it does that is that it is the instructions for making particular proteins. And those proteins make up the machinery in our cells. So this gene for APDS1 makes one protein, and this gene for APDS2 makes another protein. And these proteins work together in the cell to do something important. And what they do is they help the cells detect what's in the environment. So I told you what our immune system has to do is, walking through my day, a bacteria comes along, it could be harmful for me. My immune system needs to be able to detect that and to detect that it needs to make a response. But it also needs to be able to detect that that's a bacteria, not a virus, so it makes the right response. So what these proteins are doing is they're part of the machinery in the cell that goes from these detectors that are on the surface of immune cells that detect what's in the environment around, and they transmit that message into the cell to tell it what kind of response it needs to make. So I've got a bacteria, they detect it, they send this message. But now you can imagine that if I've made my machinery slightly wrong because the instructions in the DNA are slightly wrong, now the message that gets sent within the cell may be garbled. And the message may not get through to make the right response to the bacteria, and I'm going to get sick. So we think that's what is happening. How can we test that? Well, what we did was that we took blood from these patients. So I told you that white blood cells are some of your immune cells, so we can easily access components of the immune system from patients. And then we can isolate those cells and look at them. But what we can also do is that now there's new technology that allows us to edit the genome. So we can make mice that have that same mistake in their DNA that the patients have, and then we can use those mice to look at how the immune system responds. We can immunise those mice, we can infect them, and we can look at how the immune response changes. And then we have lots of different technologies here at Garvan that allow us to really interrogate what's going wrong with the immune system. So we spent several years actually looking at this for APDS 1 and 2, but I've only got 20 minutes to talk to you, so I can't cover it all. So I'm just going to tell you about one immune cell that we looked at, and that's the B cell, maybe my favourite immune cell. You may have heard of B cells. They're a cell type that's really important for our response to vaccination, but also to infection. And so what happens when you get an infection or you get a vaccination, there will be some B cells that recognise that infection. Those cells will get activated. They'll start dividing, so you get more and more of them because we need enough of them to fight off the infection because the bacteria is also dividing and they're getting, so we need more troops to fight it off. And what they start doing is they start making these little Y-shaped molecules that we call antibodies. And they go through our blood to all parts of our body. And they're, wherever the bacteria or the virus is that they recognise, they can bind to the surface of that. And you can imagine that's kind of blocking that bacteria or virus from kind of doing damage. But the other thing it does is that it flags the bacteria or the virus to other immune cells that then come and gobble it up and clear it out of our body. So this process is really important, particularly for those bacteria, clearing those bacteria for our body that I told you these patients have trouble and get lung and ear infections from. So we then ask the question, in the patients, is this process going on normally, or do the patients have a problem with this? And what we found was that both the APDS 1 and 2 patients had a problem changing the type of antibody. So there's a particular type of antibody you need to make so that you can flag the bacteria to be gobbled up by other cells. And these patients kind of can't switch to making that proper antibody, and so they can't flag the bacteria to be gobbled up. But there were some differences between the patients. So the APDS 2 patients, but not the APDS 1 patients, had decreased expansion of their cells and also decreased quality of the antibody. So this kind of explains to us why both patients are sick, but it also flags to us that the immune system is really complex, and even these 2 proteins that we think work closely together can have slightly different effects. So what does that mean about how we might treat these patients? Well, there's actually a drug that targets one of these proteins. And so we use that in our cultures in the lab, and we said, well, how do the B cells respond to this? Can this drug actually help correct what's going wrong in these patients? Now, this is the only real data slide I'm going to show you. So what you can see, so this is actually, we're looking at the function of the B cells to change that type of antibody they're making, the one that they need to flag the cells to be gobbled up. And what you can see here is that both the patients are worse at doing this. That's what I told you already on the last slide. But now, as we add this drug in, you can see that the APDS 1 patients recover really quickly at lower doses of drug, but these APDS 2 cells require slightly higher levels to get the same effect, and they never really get to the same level. So this suggests to us that these patients may respond slightly differently to this drug, so that as clinicians start treating these patients, they might want to keep that in mind. So that's some of the work we've been doing on APDS 1 and 2. So where to from here? Well, as I said, we hope that this information will help clinicians to treat these patients better, to give them the right treatment that will give them the best improvement for their disease. But that's true not just for these APDS 1 and 2 patients, but for all of these patients who have these rare genetic causes of immune deficiency. The problem is, some of the time we don't even know what the best treatment is. So we're doing some work with colleagues over at St Vincent's to actually look at patients who are being treated with these targeted therapies and see how they respond. So we have the evidence to make sure that we're treating them in the best way. But of course, we can't give them these targeted therapies unless we know what the problem is in their DNA. And so there are still patients out there who we're pretty sure have a genetic condition, but we don't actually know yet what that change in their DNA is that's driving that. So Shruti's going to talk to you a little bit about the work we're doing for one lot of patients to try and help them get a diagnosis there. So these rare genetic conditions don't affect that many people. So APDS is maybe 20, 30 patients in Australia, but when it does, it can have very severe impact on their life. In and out of hospital, shortened lifespan, really affects their quality of life and their family and friends around them. So it's important that we study these. But there's another really good reason for studying these diseases, and that's because it shows us the critical pathways within immune cells that help control appropriate responses. And that has implications for how we understand and treat more common diseases like autoimmune diseases and allergy, and can help us really understand that. So just in my final few minutes, I'm going to talk a little bit about how we're moving from these rare diseases to more common diseases. And to do that, I'm going to talk a little bit about when B cells go bad. So it's great when B cells make antibodies against bacteria or viruses and protect us from those things. But sometimes you can have a B cell that recognises part of us, and it also gets activated and starts making antibodies. But if they bind to parts of our own bodies, that can cause damage, and conditions such as lupus, where you get this classic rash, you can get destruction of your kidneys, you can get nerve damage, and it's very detrimental to the health of these patients. But the thing is, we know these symptoms are at the end that you're getting these bad antibody produced, and we wanted those symptoms to go away, but there's actually probably many different ways that a B cell can go wrong in the beginning so that it starts down this path. And we don't just want to treat these symptoms at the end, we want to treat what started the disease in the first place. So lupus really probably, though we think of it as one disease because people have similar symptoms, is probably actually multiple different diseases with different drivers that are making those B cells go wrong. The problem is that we don't really treat it like that. You turn up to your doctor with lupus, they don't know what the driver of your disease is. They just give you a drug that, has worked for other people that kind of suppresses your immune system. And for some people it works, and for others it doesn't. So you kind of maybe get a response like this, where the yellow people might respond really well, but the blue people don't respond well to that therapy. And then your doctor might try another drug, and that may or may not work. And, after, you can go through a series of trying a number of different drugs, and for some people, they never really find a drug that works well for them. But surely there's a better way of treating this. And what we're really hoping to do is to work out who these different groups of patients are and make sure that they get the treatment that works for them. The problem is that at the moment, we don't know how to do that. We don't know how to work out who the yellow people are and who the blue people are. So we end up in this process of trial and error. So how does this relate back to these rare diseases? Well, one thing I didn't tell you about APDS earlier is that these patients also get autoimmunity. In fact, some of them, when they first went to their doctor, were misdiagnosed as lupus because they looked a bit like that. So this tells us that this pathway might be playing a role in B cells gone bad. And in fact, in a normal B cell, you'd hope that if you saw self, that B cell gets a stop signal that says, you don't want to make antibodies. That would be dangerous. But we showed in our lab that, in fact, when we have this defective protein in the B cells, they get confused, and instead of stopping when they see themselves, they actually go and they start making antibodies, and that contributes to the autoimmunity that we see in these patients. So can we learn from these rare diseases as we try and treat other autoimmune diseases? Maybe there's a group of lupus patients or other patients with autoimmunity who also have problems that may be affecting this same machinery in their cells. They may not have a problem in the instructions, but there's other reasons why that bit of machinery can go wrong in a cell. And if we can learn from our APDS patients to identify what the signs are that someone might have a problem in this part of their machinery, then maybe we can identify the patients who would respond to drugs that are similar to the APDS patients. And that's true not just for these APDS patients with this rare disease, but there are other rare genetic diseases which also lead to autoimmunity, which may help us to identify the yellow and the green groups as well. So hopefully I've been able to show today that we can have a big impact on immune health, whether that be in these rare genetic conditions where we're hoping to diagnose more of them and treat them better, or in these more common diseases where we're hoping to really personalise the medicine so that we're treating what's driving your disease, not just the symptoms. So thank you for listening. I'm now going to hand over to Dr. Swamy, who is, as Marajeen said, a doctor and who has come to work in my lab on one of these projects about trying to understand diagnosis. So Shruti, if you would like to go.
My shoes are loud, sorry. So yes, I'm Shruti. Thank you so much for being here today and for allowing me to talk about my journey and how that then became sort of our journey here at the Garvan as part of Elissa's lab. I thought I'd start with a quote from one of my favorite philosophers, Tagore. His point towards service is really something that I've hoped to encompass in my life as I've gone on, and a lot of people here at the Garvan do, trying to find ways to make life better for so many people. So to start talking about my history, I'll start at the start, which is when I was born, and that was in India, a small little town in India called Coimbatore. At the age of 7, I remember traveling to and from school myself with a group of 8 children piled into a little man-driven rickshaw, exactly like the one in the photo, passing by all sorts of members of societies, including the haves and the have-nots. And I remember thinking at the time, it seems really unfair. There's a division between the haves and have-nots. And I think the thing that spoke to me the most was the division in healthcare. Decided at a very young age that I wanted to be a doctor. And at the age of 10, my parents uprooted their whole lives in India and brought us to Australia so that I could fulfill my dream. At the age of 17, I could not be more grateful to my parents for doing this, because I was then admitted into the Monash Medical School in Victoria. Having completed my medical degree, I embarked on a variety of things, including getting married and taking a year off to travel around the world. But at the age of 27, I did my first term in paediatrics that saw me trepidatious and incredibly anxious about treating these young little people. But since starting that, I never turned back, and I really do feel that paediatrics chose me. At the age of 29, when I was studying for my exams, I was fascinated and blown away by the immune system and how it underpins absolutely everything and every organ system in our body. And it spoke to me. And so I decided to subspecialize in paediatric allergy and immunology. At the age of 37, 2 children, multiple part-time years later, I finally became a consultant in the field of paediatric allergy and immunology. So through my clinical journey, I've seen several patients, spent an inordinate number of hours with them, have worked at 8 different hospitals, and keeping in the vein of hoping to bring equitable care, in this case geographically, for those who are geographically possibly disadvantaged. I'm very honoured to have set up 2 allergy and immunology clinics in some regional and remote areas in Australia. So there are some clinical insights, and I won't go into too much detail, but a few key points that I think have come through all my years of training and working as a doctor. And one of those things is that simple changes can have big impacts. And whether that's, as Mara Jean showed us, a bit of Ventolin to help your breathing, or a child who's waking up every night with severe eczema, who then gets the basic management changes and is now a different child, as their parents often say. Helping children during their young years means you change their world. You absolutely change not only their world, but the world of their parents, their carers, their siblings, and their little microcosm as a whole. One of the first things that struck me is that chronic disease does not just affect adults. These very little people also get very big diseases, and that carries through with them, often leading to significant degrees of disability as their little lives progress, or in some very unfortunate instances, mortality. I have found the science behind the immune system incredible. As I said, it underpins so many other organ systems that we have. It affects our lungs, it affects our brains, our nervous system, our heart, our joints, our skin, as you've seen with the lupus patients, and our blood, and variety of different organ systems. And if I've missed anything, I'm sorry to the body parts that I've missed, but the immune system is probably implicated in those as well. And one of the things that I've discovered more as I've neared the end of my training and transitioning into my world as a scientist or a researcher is that treatment without evidence base can do more harm to children and adults and anyone receiving treatment than good. So this brings me to the patients that I'd like to talk to you about, and we'll call them Alice and Joshua. They were incredibly young children in their first few years of life who were siblings. When they first came to the hospital, they presented with severe eczema, They could not sleep through the night. They were itching to the point where there was blood on the sheets. And the parents were distressed every morning looking at this thinking, what is happening to my children? They had very early onset lung disease, again within the first few years of their lives, which meant that they had decreased exercise tolerance. They couldn't run around and play with their friends. They were chronically coughing, having to stay home from school, missing loads of education, really all the things that you would expect a normal child to be doing at that age. They were incredibly short, so they looked multiple years younger than they actually were. And again, going to school, this meant that they stood out apart from their skin and apart from their chronic infections, they were incredibly small. And they had multiple food allergies. And if anyone knows anyone with food allergies, that means, lots of vetting of parties that they're going to, birthday parties, saying you've got this special food and you have to eat this food. So they had a whole bunch of significant things going on. And the parents, as you can imagine, as any one of us would feel in that situation, were lost and helpless and wanted to have hope and wanted to understand what was going on with their children. And if there's anything that we can do for their children, apart from antibiotics, which is the only main way we could manage, for example, their lung disease at the time. So I might just show you images of the patient themselves. So this is what a normal lung would look like on a CT scan. And at the first few years of life, this is Alice's lungs. So as you can see, even just looking at between those two lungs, one is incredibly diseased compared to the healthy. So in the pursuit for an answer, the patients had several investigations done, including blood tests, samples from their lungs, biopsies, and a whole array of genetic tests. And then we arrived at a possible genetic cause. The reason we thought it was possibly implicated is even though it was a novel gene and it's never been described to affect patients in the way it has these patients, there was enough data that we know about the protein that's made by the gene, as Elissa was saying, that if it's not working properly, we expect it to have issues all over the body. But what does this mean specifically in terms of these patients? So then began the journey where the family had very reasonable questions, including, does this provide an answer? What does this mean for our children and really their children? How will we know if this is actually the cause and we're not running down a rabbit hole that's not actually real? And it brings me back to my point of the fact that treatment without evidence, you do more harm than good. So we needed to find the evidence, and that's where my journey came along and brought me to what I'm now focused on, which is translational research. Translational, basically translating from the bedside of the patients to the bench at the lab, back to the bedside of the patient with treatment options, with answers, with possible answers, or even at the very least, a yes or a no. The question is, how do we interrogate this further to provide some real answers for the family? And as Elissa mentioned, there's a lot of ways we can look at the immune system, including as simple as a blood test, where we can separate the white cells and look at them. The problem with these patients, of course, is their lungs are so affected, and we can't really get samples of their lungs multiple times in order to assess what's going on. So the very clever people at the Garvan helped us create a mouse line with this particular genetic variant incorporated into it. This way, we have the ability to explore the lungs of the mice, to assess the nutritional status, and to assess their immune system in a little bit more detail. It would also help us hopefully identify therapeutic options if this gene is indeed looking like something that's implicated in these patients. We can then take that back to the bedside of the patients. So what have we found so far? As I mentioned, one of the most striking things that you see when you first look at these children is how small they really are. And actually, one of the striking things that I found in these mice is actually how small they are compared to their unaffected counterparts. And really measuring multiple different mice over time, it looks like they are, in fact, much smaller than their unaffected counterparts. The reason that's exciting is because we've got a particular gene that we don't know if it's involved in the patient's phenotype or how they present. But here we are starting to have glimpses of the fact that it might, in fact, be this gene that's responsible. Might. So what about the lungs? Diseases of the immune system, and specifically the lung-specific immune cells, can lead to a resulting lung disease that's very much like the children. And there are these guys over here. And I know Elissa's talked about T and B cells, but we've also got what's called macrophages, and they live in your lungs. They are responsible for keeping you healthy from a perspective of infections, but also look after the lung integrity and making sure that the lung heals, repairs, and maintains itself without going into overdrive or underdrive. And again, what have we found looking at the lungs of our mice? One would postulate that if these macrophages are in fact associated with a defect in these patients' lungs, then you'd have to see either a reduction in the numbers of these macrophages or a problem in their function in order to potentially explain the patient's phenotype. So we set out to explore that within our mice. And we found that actually these mice had significantly lesser alveolar macrophages, these particular macrophages, compared to their unaffected counterparts. Again, that adds a little bit more, just one more block to the story that we're trying to understand for these patients, and potentially undiagnosed patients across the world who have not been identified because there's not been a knowledge about this particular gene. So what does this mean? And I think my time in research has taught me, if it's taught me one thing, it's that it's raised more questions, really. Is this a defect to do with the lungs itself or to do with the cells in the lungs? And that matters because if we just replace the cells by transplanting the patient's cells with healthy donor cells, will that be enough to fix them? How will this discovery affect the world of these children and their parents? But equally, talking about transforming immunology, how will this affect the world of immunology and genetics as we've come to understand it? And do we have a whole new gene that's implicated in patients that have previously been undifferentiated? It's early days, but it's incredibly exciting days for us to be able to take something back to the world of the patients. So I'd like to thank you for allowing me to present my journey today, and I hope this brings us to a clearer future as we head forwards. Thank you.
Wow, am I right? Can we get another round of applause? So look, I've got some questions of my own, but this is an opportunity for us to open up to the audience. So if anyone here in person has a question, please just pop your hand up and we'll get a microphone over to you in just a moment. Also for our lovely people in the Zoom room, there is a Q&A function. Please feel free to pop in any questions or comments you may have. I will just gently remind everyone that for obvious reasons, we cannot provide individualised clinical advice for you. So if you are motivated from this conversation and would like to actually explore your own immune health, I would strongly encourage you to visit your GP and get a referral to a clinical immunologist. But I think we do have one question down here, so we'll just grab the mic, please, Caroline.
Thank you for the talk. It was very interesting. I just wonder if you're actually considering other aspects of the children or the immune system. So children are born with a pretty well perfect immune system, but nowadays I think they can get up to 47 vaccinations. I'm just wondering if each vaccination weakens their immune system. And have you considered outside factors like with the parents being vaccinated, because the stillborns are increased by about 50% in my area after the vaccinations for COVID. And also the fact that there are so many chemicals around houses. So I ended up using one of those loo things and getting incredible migraines, which I don't get. So I just wonder about children being that much closer to the toilet.
Am I working? Yeah, look, I think it is interesting. We know that the environment can interact with disease. I think in the case of these kids that Shruti is talking about, well, all of the environments around us have changed and most people aren't that sick. And So I think there's definitely an impact of environment on our immune health. Um, you know, I don't think there's actually good evidence that vaccination is making us worse. I think it makes us better. It gives us protection. There are kind of the, odd occasion where people have bad responses. Um, but that's not generally, I think, suppressing our immune system. So I think when we look at these really severe diseases, we do know that we kind of look at them and say, this is so severe, we think it really must be something genetic that's going on. But one of the big areas of research is actually understanding how the environment interacts with genetic things. So actually one of the things that Shruti mentioned briefly with her project is actually we're looking at how diet impacts this genetic mutation. So we know that for these patients, actually, one of the things that we see in the macrophages is that they look kind of a bit fatty in the lungs, which suggested to us that actually there may be a component of kind of the way that they're able to process fats that may be impacting their disease. So we're actually looking at, well, does a fattier diet impact things? And we know that in other immune conditions that things like diet can have a huge impact on the immune system. I don't know if you want to.
No, I think you covered everything very well. But yes, and even in terms of how you were talking about pregnancy and the outcomes of pregnancy, um, there are multiple different things that are happening as time's going on, including use of antibiotics, including use of, as you say, hand sanitization, all those sorts of things. I think the issue we face is it's not unifactorial, it's multifactorial. And I guess the evidence so far has supporting our immune system as much as we possibly can with the immunizations so that patients like this, where some parts of their immune system is not working, stand a fighting chance with the bits of immunity that we confer by giving them the immunizations that we do.
Excellent, very helpful. Thank you so much. We've got some fantastic questions coming through online. So the first one here is for you, Shruti. What is a potential cause for a reduced production of macrophages? And could there be any link to an aberrant protein? I think we've got a scientist in the Zoom room.
That's a great question. Thank you, sir or madam scientist. So that's a fascinating question, and I won't bore everyone with the details of every aspect of it, but suffice to say, we know with these particular type of macrophages, there's two populations. One that that you're born with, that you are born with, that live in your cells and multiply in that particular space to regenerate as they get knocked down by infection or smoke or viruses. And there's ones that come from the bone marrow that then replace these ones and provide this ongoing pool of macrophages that's contributing to the ones that are already in there. So really the more questions, which I didn't put up there, is which of these macrophages are struggling in this particular setting? Is it that the ones that are already in there are just not able to multiply and regenerate as time's going on and different insults come along? Or is it actually a defect in those bone marrow derived ones, the ones that are coming from somewhere else? Are they just not able to get into the space? Which brings us back to, is this a lung defect or an immune cell defect? More questions, unfortunately.
Brilliant.
But ones we're hoping to answer.
Thank you. And clarification from Bev, ex-nurse. How much do we love our nurses? Bravo. A question for me. If I understand correctly, autoimmunity significantly impacts women at a higher ratio than men. Why?
Do you want me to take this one? The short answer is we're not really sure. There are various hypotheses, and actually probably there's multiple aspects that contribute. So one is that there's some impact of kind of sex hormones that we know that they can impact immune cells and make them change. There's also, if you know much about genetics, women have two X chromosomes, men have an X and a Y. Because we only really need one X chromosome in women, kind of one gets turned off in our cells so that we only have one. Generally, we only have kind of one that's functioning and leading to protein production. There is a hypothesis that in women, maybe some bits of that X chromosome don't get turned off properly, so we end up with higher protein production of some of those proteins that are encoded on the X chromosome, and that they may make us more reactive to autoimmunity. But also, it comes with a benefit though. We may get more autoimmunity, but generally speaking, often we have stronger immune responses. So for example, something like COVID, women had better survival against COVID than men. So we might get more autoimmunity, but maybe there's a trade-off for that as well.
And men get more asthma and allergic diseases as well, and allergic rhinitis. So
we share the love with these diseases. That's good to know. Good to know. Another question that's come through online. With known genetic mutations, 1, do you have a recommended panel of tests? 2, are you looking at nutritional therapies, not just diet, vitamin D and beyond? Yes, just 2 questions. Who'd like to go first?
okay. So I think the first question was about non-inherited, so somatic, so variants that are then sort of arise beyond birth and in your cells of your body. Yes, absolutely. If anything, that the last probably 2 years, probably a bit more, has taught us that there's a huge amount of literature or things that we don't know about in terms of uninherited immune disease due to a genetic variant. And that's things that are being explored at the moment. In terms of further testing for that, it really does depend very much on whether you're in a public setting, a private setting, which hospital you're at, which state you're in, and what kind of access you have to genetics. So I would definitely encourage you to reach out to your local genetics service to see what options there are for you and any patients that you're worried about. And I think the second part of that question was
nutritional therapies,
yes,
not just diet.
So look, nutritional therapy. So for example, we know that vitamin D replacement, zinc replacement can actually help skin, oral mucosa. Yes, there's absolutely all those things that we're aware of. And I'm not sure if you're talking about in our particular patients, but that's certainly things that we did optimize going into their care. But I think this is a very fledgling project at the moment. And I think we'd have to look further in detail as to how some of these things impact the metabolism and the health of the patients as a whole, or the mice as a whole at the moment.
I think we've got another question from the audience.
I was just wondering about SLE. When I was nursing, I had a colleague with SLE. She was in her 40s, and I just wondered how the treatments changed and the incidence of SLE at the moment.
I can take that. There was a big study recently. I'll deal with the first bit first, the last bit first, about incidence of things like SLE or lupus and other autoimmune diseases. And there is some evidence that they are increasing. But there's also a bias that we also get better at diagnosing and recognizing those things. So but there does seem to be some increase in those things. In terms of therapies, there are several new therapies that have come onto the market in the last few years. But as I kind of referred to in my talk, they work for some people, but not for others. And we still aren't clear who they will work for and who they won't. So there's still really a large element of trial and error as people go through different therapies. But there are a couple of new therapies, so there are more options. It's just a matter of working out who works best with which therapy. And there may still be some people who none of those therapies will work for, and we still need to develop further therapies that work better.
Thanks, Elissa. We've got a few more from the audience, but there's a related question here online that I'm just going to jump to first before we come back to the audience. There have been some recent successes in treating autoimmune diseases such as SLE with CD19 targeted cell therapies. Is this a consideration of your research?
No, we're not currently looking at targeting CD19. So CD19 is a molecule that's expressed by B cells, and what they're actually referring to is people had made what we call CAR T cells. You get basically you generate T cells that will come, you give to someone, and then they go around and they'll kill all your B cells. So people have done that. And for a handful of patients, I think it was 4 or 5, that worked quite well. Clearly, though, knocking out all your B cells is problematic, and it's really a last line of treatment that you want to go down, because we know that our B cells are important for protecting us from infection. And in fact, if we wipe them all out, we know that people can have then susceptibility to other infections. So some people are working on that, and I think maybe for some people that will be like a last option when nothing else works. What we're actually hoping to do is have more targeted kind of precision therapies that hit those drivers rather than just bulk wiping out all our B cells, which may have bad outcomes in other ways.
Excellent, thank you. David, hi.
Hello, good morning. If it's still morning, that is. My question actually goes into 2 parts. First part is when we were kids, most of us was playing outside in the yard in the dirt. So that's an environmental factor. Have you, in your research, differentiate between the younger and the older generations? And if you have, is there anything showing that could reflect this autoimmune disease? That's the first part.
Should we go with that? Should we go with that quickly, David, and then we'll come to the second?
Okay.
That's a very astute point, and I think it does point to the multifactorial aspect of what's a rising health burden and health concern at the moment. Absolutely, our research at the moment is not looking at that specifically, but there is research, as you probably know, looking at exactly that and wondering about whether the clean theory, are we too clean at the moment to allow our immune system to be exposed to the right stuff to make sure it doesn't attack ourselves or attack things that are not harmful like peanuts and pollens. While it's not a causative link that's been identified, I think it is certainly a confounding factor amongst several other factors, and it's something that points that environmental factors have a huge effect on some of the things that are going on. But equally, one of the things that's quite interesting now that's coming out is that we know that patients, for example, or parents, sorry, who are born overseas and come to Australia and have children in Australia, these children are actually at a much higher risk of developing some of these immune-mediated diseases than their Caucasian counterparts. And so, again, the question about environment and genetics, I mean, it's all quite a mush at the moment, but suffice to say there's a lot of stuff that goes into it, including the clean theory.
And David, before we get to your second question, because we've got a number of these questions coming through online around genetics and heritability, can we just expand upon that briefly in terms of, can a parent's DNA be damaged through environmental exposures, somatic mutations, and then pass that on genetically to their offspring?
So a somatic variant passing on to the offspring is incredibly small. It's the same as the population generally. The question about whether environment affects parents' genetics, I mean, while what we understand about, for example, we know that allergies can run in families. Invariably there's, the mother or father who've got hay fever, and then there's eczema, and then there's food allergies, et cetera, and the cycle continues. Often it's to do with polygenetics, where it's not just one gene implicated, whereas with multiple genes, but in combination with environment, where epigenetics comes into it, like the families who are born overseas and come here. And there's a whole bunch of different things that go into kind of defining the parent's phenotype or clinical presentation that then allows the children to either inherit or live in the same environment or move to a different environment that their epigenetics is not quite sort of fit with. So I don't know if that answers the question, but yeah. Did you have anything to add to that?
I guess just to say that there certainly are cases with these rare variants where you'll get an individual who they're the first person in their family to have that genetic condition. So they've, developed it. I guess the mutation probably happened in the sperm or the egg before they were born. They then have that condition and then they pass that on to their children. So we do see that sometimes that, a mutation will arrive because all of us, all our cells, every time a cell divides, every time that you have to replicate your DNA. And you can imagine if you have, it's billions of letters of the code in your DNA. You can imagine that if you're copying that out over and over again every time a cell divides, that you can get mistakes. And a lot of the time, those mistakes will make no difference. Sometimes they will cause disease. And if it happens in a sperm or egg, then it can be passed on to your child.
Part 2.
Dun, dun, dun. The individual, is there any way they could actually enhance the immune system other than drugs?
Ooh, this is one of my favorite questions. And I think it's one of my favorite questions because I don't know if you've ever watched an ad for a supplement or something like that, and it will say, boost your immune system. And I think, I'm an immunologist. How are they measuring that? I don't know how to measure that. How are they measuring that? So I think that's one of the complexities. Like we know that things like, if I'm malnourished, my immune system will not function as well. But how do I measure like that it's at its optimum? And it may be that what's optimum for one thing is bad for the other. Like, so for example, we were talking about females and their propensity to autoimmunity, but then they're also better at fighting off some infections. So yes, I can improve my immune system by being generally, good nutrition, that kind of thing. But how do I know when it's at my optimum? How do I know that I'm not increasing the wrong bit of it and it's going to go wrong somewhere else? And that's, I think, one of the things that we've really got to work at is actually how do we measure immune health well so that we can look at your immune system and say, these bits are working well, those bits aren't, what can we do to fix it? But we're actually still have very blunt tests in the clinic for measuring that and to knowing when it's going well. So that's kind of a roundabout answer to your question. I don't know, Shruti, if you have more specifics.
No, I don't have specifics, but one other point I'd like to make is so if you're talking about supplements, for example, we know there's a whole bunch of other things in the body that go into whether those supplements make it to the right places, etc., including your gut microbiome, and things like that, which is, fairly intrinsic to you. I mean, things can affect it. But again, it comes back to the question of what are we delivering, how much is getting there, and what effect is that having, and how is it measurable? Tricky.
Fantastic. Now, I am very sorry to say that we are right on time. However, for those of you in the Zoom room that we were not able to get to you, please do pop your questions through in an email and we'll get some responses to you over the coming week. For those individuals in the room, please, I think we've got the profs for a few extra minutes. So if you're happy to stay and just ask those questions one-on-one, that would be great. Again, I'd love everyone to put their hands together for this incredible presentation. If you've been inspired by today's session and you're not already a member of the Garvan family, please do consider signing up in an ongoing way. Make a donation if you can. Advocate, tell your friends all about us and the incredible work that's happening here. The next and final public seminar for 2023 will be on Friday, the 8th of December. So please do save the date and RSVP to come along to that one. The topic is currently to be confirmed, but it's going to be fabulous, I can guarantee it. We also have a new podcast series that's just launched, and Elissa is in the latest episode. So if you like learning more and if you like podcasts, please do jump onto the Garvan website and you'll be able to find our podcasts there, and a really exciting session from Elissa continuing on this very theme today. So thank you so much, everyone, for coming. It's been so lovely to see you. Thank you for everyone in the Zoom room, and have a wonderful day and a great weekend.